As I said in the previous video, you don't need to have a lot of prior knowledge of chemistry and biology to follow this course, as I will present all the necessary basics whenever necessary. So in the next few slides, we are going to have a closer look at what proteins are. Proteins are large biomolecules responsible for a vast range of functions in all living organisms, including catalyzing metabolic reactions, replicating DNA, the genetic code of all cells, responding to stimuli, providing structure to cells and organisms, and transporting molecules from one place to another. Proteins are made up of long chains of amino acids, and there are 20 different amino acids that can be present in such a chain, and each amino acid is a small molecule. On this slide, you can see an example of an amino acid, the amino acid alanine. You can see that the amino acid is made up of different atoms bonded together. And later in this course, we will have a more detailed look at what small molecules are and how the different atoms are linked together. But for now, you just need to know that amino acids are small molecules. So proteins are long chains of amino acids, and successive amino acids are linked together by chemical bonds that are called peptide bonds. Such protein chains typically consists on average of a few hundred amino acids. And when a protein amino acid sequence is built, the protein sequence does not remain a linear sequence, but instead it folds into a 3D structure. And this folding occurs because of the attraction and repulsion between different parts of the amino acids in the chain. For example, some of the amino acids like to be closer to water, which is typically present in the environment of the protein, and some parts try to avoid water. Because of these preferences, the protein chain bends and twists so that the water avoiding parts are more on the inside, away from the water, and the water loving parts are exposed to the water on the surface of the protein. This process is also influenced by bonds and interactions between amino acids that don't have to be close or next to each other in the linear sequence. All of these forces together cause the protein to fold into a specific 3D shape. And this shape is very important because it determines the function of the protein. To give you a better idea of how protein amino acid sequences are made, I'm showing you a nice visualization of this process from a YouTube channel called Sprong Studio. Protein amino acid sequences are produced by ribosomes, which are also large macromolecules, and you can see a ribosome here in the right border of this video. And in this video you can see the process of protein production and folding. These ribosomes follow the instruction for the amino acid sequence that is encoded in a molecule called the messenger RNA, or short the mRNA. So originally, the DNA contains the instructions for every protein sequence in a cell. And when a specific protein is needed, the cell makes a copy of the relevant part of the DNA with the instructions for the protein and converts it into mRNA through a process called transcription. And then the ribosome makes the amino acid sequence that is encoded in this mRNA in a process called translation. And as you can see in this video, once part of the sequence are produced, the protein begins to fold and you end up with the 3D structure of the protein. As I said, if we have different protein amino acid sequences, these will fold into different protein 3D structures with different functions. And in every cell, there are thousands of different proteins that all have different functions. For example, even in a simple bacterium such as E. coli, which is an organism consisting of only one cell, there are approximately 3,000 different proteins present and a total number of approximately 10 million proteins in each cell. All of these 3,000 proteins have different structures and functions. They are, for example, responsible for producing all the relevant molecules required for cell growth, for taking up molecules from the environment, for producing new proteins, and for giving the cell its structure. And in all of your human cells, for example, your skin, blood, or brain cells, there are even more different proteins present, approximately 10,000 different proteins in each cell. The images on the slide hopefully give you an idea on how diverse the resulting 3D structures can be for different protein sequences. On the next slide, we will look at some different functions that proteins can have. One important and large class of proteins are enzymes. Enzymes are proteins that can catalyze chemical reactions, which means they can massively speed up chemical reactions up to 1,000,000 times or more. Most chemical reactions that take place inside biological cells could also take place without enzymes, but they are much less likely to happen, and it would take far too long for them to occur. In this image, you see an example of an enzyme, lactase. Lactase is an enzyme that catalyzes the splitting of lactose, a milk sugar, into smaller sugar molecules, glucose and galactose. This means that the enzyme lactase helps to break down lactose molecules, a sugar found in milk and dairy products, into smaller molecules that can then be further utilized by other enzymes. The lactase enzyme has a binding pocket in its 3D structure where lactose can bind to the enzyme. And this helps to break down lactose into molecules that our bodies can easily use. For example, for people who are lactose intolerant, this chemical reaction of breaking down lactose into smaller molecules does not happen fast enough because the lactase enzyme is not produced often enough and hence the cells cannot use the milk sugar as an energy source. And as a result, undigested lactose remains in the gastrointestinal tract, causing a variety of symptoms such as stomach pain or diarrhea. The small molecules that an enzyme can bind to and can convert are called substrates. There can be one or more substrates in every enzyme catalyzed reaction. And the transformed molecules that are the result of the chemical reaction are called the products. And like the substrates, there can be either one or more products. In this example here, we have two products, but it could also be the case that we have an enzyme that only changes the structure or conformation of a single substrate, and this would then result also in only a single product. It is also important to note that the enzyme is not altered in this process. This means that the enzyme binds to the substrate, it transforms the substrate into products, and then it releases them, and the enzyme has not changed during this whole process, which means that it can again bind to a substrate and again catalyze the same chemical reaction over and over again. In every cell, there are many different enzymes that catalyze different reactions. In humans, for example, there are at least 3,400 different enzymes with different functions. On this slide, I will list some of the major classes of proteins to give you a better picture of what proteins can do. So the first class is enzymes, and we already have seen an example of enzymes on a previous slide, so I will not further talk about enzymes here. Another example of proteins are transport proteins, which are proteins that can transport molecules across cell membranes. For example, the cell membrane separates the inside of the cell from the outside of the cell. And cells need to be able to take up molecules from the environment and also to be able to transport molecules from the inside to the outside of the cell. And this is accomplished by transport proteins. The next example is regulatory proteins. These proteins regulate the activity of genes and gene expression to ensure that the right proteins are made at the right time and in the right amounts. In this example, you see the tumor suppressor protein p53, and the task of this protein is when DNA damage is detected, this protein can stop cell division and activate other proteins that are involved in DNA repair. And The last example of a class of proteins that I will show to you are structural proteins. These proteins provide support and shape to cells and organisms. And one example is the protein collagen, which looks like strong long cables or ropes. And we find collagen, for example, in our skin, bones, and muscle cells. And for example, it helps to give your skin strength and elasticity, which allows the skin to stretch without breaking and then to return to its original shape. Okay, so hopefully now you have a rough idea about what proteins are and what their functions can be. Now we want to answer the question, how can we store information about the protein? We have seen that proteins are sequences of 20 different amino acids. And we also know that the amino acid sequence of a protein determines the protein structure, which determines the function of the protein. So in principle, the amino acid sequence contains all the information we need to have about a protein. So we can represent the protein by its amino acid sequence, which is the most common way to store protein information. The amino acid sequence of a protein is usually represented by a sequence of capital letters, where each letter encodes an amino acid. This is very compact and storage efficient since you only need to store one string for each protein. Here you can see on the right all 20 amino acids that can be present in a protein. In this example, we would have a protein starting with methionine in the first position, leucine in the second position, and so on. Since this amino acid sequence already contains most of the information about the protein structure and function, this is the way we will usually represent proteins in this course. We can store protein amino acid sequences in FASTA files. The FASTA format is a text-based format, which means you can open and edit it with any text editor. Each entry in a FASTA file starts with a greater than character, followed by a description of the protein in the same line. In this example, on the slide, you can see that it simply says sequence 1 for your first entry and sequence 2 for the second entry. But in principle, we could add any information we want about this protein to this line. In the following lines, we have the amino acid sequence of the protein, and each line usually contains no more than 80 amino acids, but this is just a convention and can be different. And all the amino acids until the next entry that starts again with a greater than sign belong to the same protein amino acid sequence. So as you remember, the linear amino acid sequence folds into a 3D structure. So you may wonder, why do we not encode the protein 3D structure instead of the linear protein amino acid sequence? Since we know the 3D structure defines the function of the protein, so maybe we can get this information more directly from the 3D structure. And if the protein structure is known, we can do this. We can determine the protein's 3D structure either by experimentally determining it, which can be difficult and time-consuming, or by predicting it using deep learning models such as AlphaFold or ESMFold. We can store this 3D structure using PDB files. PDB files are also text-based files that contain the 3D coordinates of all the atoms in the protein. In this example, you can see that valine is the first amino acid of the protein in the protein amino acid sequence, and you can see that all the coordinates of the atoms in the valine amino acid are stored. And we do this for every of the amino acids in your whole sequence. So in the end, you store all the 3D coordinates of all the atoms in your protein. This is, of course, much more memory intensive, but it's also much more accurate and stores much more information than simply using the amino acid sequence of the protein. However, currently the best methods for numerically encoding information for large numbers of proteins are based on the protein's amino acid sequence. And so we will usually represent proteins using their sequence.